Well, thank you for being here. Today I'm going to give a talk on the genomic and chemical basis of sexually selected traits in mannequins, insights from hybrid zone and feather analysis. So much of the research on mannequins focuses on the fact that their life history and evolution history are shaped by strong sexual selection. The mannequins have a leg mating system with male gathering legs and performing elaborate courtship dances in display course. Other specialized traits include elaborate social organization and special morphological adaptation, such as an ability to make loud non-vocal sounds during courtship displays. Of course, the other thing that stands out the most about mannequin is that the male mannequins possess colorful plumage compared to the females, which tend to be just greenish in color. Now, this coloration could be caused by melanin, feather structure variation, and carotenoids. Here I've listed some carotenoid compounds that are responsible for the yellow to red color found in the males of these four mannequin species. There is a range of carotenoid compounds known in mannequin feathers out of a total of about 600 known to occur naturally. Some of these compounds are naturally found in their diet, that means they take it from the plants, while others need to be metabolically transformed by the birds. So here the big questions are how are different dietary and metabolic pigments get co-opted to produce the plethora of colors we see in this group of birds that are under strong sexual selection and are primarily frugivorous, and also how do various genes and biochemical pathways evolve during the diversification of this group. Now, with genome sequencing and the use of various avian systems, research groups are finding out genes are responsible for color differences in birds. Now, many studies looked at either melanin or carotenoid colorations, but a few do look at both. Some of the melanin-related genes and systems are shown to the left. You have the famous MC1R gene and the agouti signaling protein. On the right are some of the genes that control carotenoid color differences. Examples of this include the cytochrome P450 gene family and the scavenger receptor gene. Now, to begin to answer questions about color evolution in a group that's under strong sexual selection and the types of pigments that are involved, we'll start by looking at the study system based on the genus Manicus. The genus Manicus is shown here with the red box in this new mannequin phylogenetic tree. So the results I'll show you today are based on two sets of complementary approaches. We use microscopy, spectroscopy, and liquid chromatography method to show what chemicals underlie color differences among Manicus species. We also sequence genome and used genome-wide markers to determine the genes and pathways that are involved with color differences. Now here's a quick background introduction to the group and the biogeography context. So in the Manicus genus, there are four recognized species. In this map, you can see that primarily to the east of the Andes, you have Manicus manicus, the white-bearded mannequin. So he has a white neck, gray belly, and black wings. And west of the Andes, you have the other three species, as well as a western Andes population of Manicus manicus. Now, the three species to the west of the Andes vary with respect to their neck color and their belly color. So here we have three species, the white-collared mannequin, WC Manicus candii, the golden-collared mannequin, the Villinus, and the orange-collared mannequin, Orientatus. Now, if you pay attention to the tree on the right, the white-bearded mannequin population west of the Andes, over here, is actually sister to Manicus villinus, which makes it appear that the yellow coloration is a novel trait and may be spreading within the species. In our study, we looked at what color compounds are responsible for color differences between these four species. We also made use of a special hybrid zone between the Villinus and Candii, so the two species shown with the asterisk. So hybrid zone is in Panama over here. We use this hybrid zone to study the genetic basis of color variation. So here's a zoom-in look at the hybrid zone in Panama. At this hybrid zone, the yellow color and olive green belly of Villinus is introgressing asymmetrically into a Candii population, such that there is a Candii population that looks like Vitellinus but is genetically Candii. So I shall call this population here the golden colored Candii. However, the introgression of male plumage trait seems to stop at this river here, the Rio Changanola. And west of this river, the Candii looks normal, and I will call them normal or parental Candii. In our genomic work, we resequenced the genomes of 24 birds, 2 from Candii far away from the hybrid zone to the west, 10 normal Candii located just west of the river, and 10 golden colored Candii collected from east of the river, and 4 parental Villinus from further out to the east. So here are some of the results. First, the results from chemical analysis. Using high-performance liquid chromatography, we see that differences in yellow coloration in the neck across species is simply due to different concentration of the dietary carotenoid lutein. So this plot here shows the concentration of lutein in the feathers of the neck. So that's along the y-axis, with species roughly arranged by having the least amount of yellow to the left and the most amount of yellow orientators on the right. So here you can see that there's no lutein in Manicus manicus and Candii. The Villinus and the golden colored Candii both have golden neck feathers, and the amount of yellow or lutein in neck feathers is highest in Orientators. To your right is an exemplar HPLC output. The 6 known standards elute at different times, and these are shown as a shorter yellow or red peaks. The carotenoid extracted from the neck feathers of Orienticus or orange colored mannequin eluted as just one single peak, number 7 in black, and it matches standard number 3, which is lutein here. Now for the belly feathers, we again detected only one carotenoid compound, lutein, in all species. Again, the birds are arranged by belly color. Manicus manicus has a gray belly, and he has no yellow whatsoever, and as such, there's 0 lutein in the feather. The Candii has a pure yellow belly, and the rest of the birds have olive green belly. So although the concentration of lutein varied by quite a bit, all species other than Manicus manicus have some level of lutein in their belly feathers. Again, on the right is an example HPLC plot for Orienticus, with the peak number 7 coming from the belly feather extract, and it matches lutein very well. So what is causing some of the other species to have olive green belly? The answers came from our microscopy results. So here you can see images of collar and belly feathers of 3 species. You can see for Manicus manicus on the lower left that the gray belly is due to the deposition of melanin in the feather barbules. You can see dots of melanin on the barbules, while the white neck feather has none of this melanin in the barbules. The olive green belly of the Villinus is caused by a combination of yellow feather barbs in the middle which is colored by lutein, and melanin in the barbules. The golden neck feather has no melanin as expected. Further, the barbs are thicker than what you see in Manicus and are missing their barbules. For the Candii, again, the yellow belly feathers are pure yellow. The barbules are not missing, they're just not shown in this picture. The neck feather is translucent, just like that in Manicus. So moving on to genetic results. We have sequenced 24 birds, and sequences of these birds are mapped to a reference genome of the Vitellinus. The Vitellinus reference genome is a hybrid assembly using PacBio data and Illumina data. The reference genome of the Manicus Vitellinus itself was scaffolded against the high-quality reference genome of Chiroxifia, the lens-tailed mannequin, create chromosomal level scaffold. In the end, after mapping, SNP calling, and filtering, we have roughly 20 million SNPs for the 24 birds from the two species. Now, to make sure that we really have plumage traits that are introgressing from Vitellinus into a genetic background Candii, we first ran principal component analysis to look at population genetic structure across a hybrid zone. Now, this PCA plot shows population A, B, and C. These are Candii populations from both sides of the river. They cluster together on the left side of PC1. And Vitellinus population D is on the right side of PC axis 1. And PC5, which only explains a small amount of genetic variation, separate the two Candii groups from the two sides of the river. So A and B Candii are normal Candii from the west side of the river, and C are the golden colored Candii. Now, we then conducted demographic and population divergence analysis using site frequency spectra. We look at the divergence history between three populations: the normal Candii, the golden colored Candii, and Vitellinus. The demographic model that fit the data best was one with asymmetrical gene flow between the populations that are adjacent to each other geographically. So this plot on your left shows the split times between population in the y-axis, and the width of the rectangles roughly represent effective population size. So you can see here that Vitellinus diverged from Candii about 1 million years ago, and the two Candii populations separated around 130,000 years ago. So gene flow rates, which are shown as arrows, tend to be low, in the order of 10 to the power of -5 to -7, except in one direction, which is from the normal Candii west of the river into the golden colored mannequin east of the river. This gene flow rate is about 3 times higher than in the opposite direction. Now, this probably explains why the golden colored Candii can still maintain an overall Candii genomic background despite ongoing genetic contact with Vitellinus. Next, we conducted sliding window 10 kb windows analysis across the genome to find out what genomic loci are associated with plumage color variation. So we did this analysis using two approaches. The first one is to find signature of introgression from Vitellinus into Candii, golden colored Candii, using two metrics. The first one is FD, which is a version of the ABBA test made for small genomic windows. And the second metric we use for introgression analysis is relative node depth, RND. RND is simply taking the divergence between the normal and golden colored Candii divided by divergence between golden colored Candii and Vitellinus. Now, a value that is greater than 1 means that the golden colored Candii and Vitellinus shared a more recent ancestor for that window. The second set of metrics we use are differentiation statistic between the normal candii from west of the river and the golden collet candii. To look at differentiation between windows, we use two metrics, net dxy and FST. So we would expect that the genetic loci that associate with plumage traits introgressing from Villinus into golden collet candii to show high levels of introgression and also high level of differentiation between the two plumage forms of candii. So here are the results for the sliding window analysis. Each dot here represents one 10 kb window, and the alternate colors show different chromosomes, including the Z chromosome at the end on the right. So the plots for the two introgression metrics are on top, and the two differentiation metrics are at the bottom. A dot that is red tells us that the window is a 99.9 percentile outlier for that metric. So I've listed a list of a bunch of genes that are inside or adjacent to windows that have strong support, meaning that they are supported as outliers by multiple metrics. So these genes are listed have a number of functions, including those having to do with melanogenesis and carotenoid metabolism. Some of them also have some housekeeping functions. So I won't go through all of them, but highlight a few prominent peaks. Now in chromosome 1 on the left is a solid carrier protein in the peak over here. A similar protein that associates with melanin color differences in seed eaters was found in another study. In chromosome 2, EIF3E is a gene that has to do with initiating protein synthesis, and next to it, we have the gene RSPO2 that's involved in wing signaling pathway. Further downstream of chromosome 2, we have ANGPT1. This is a gene that encodes for glycoprotein that functions in vascular development and angiogenesis. The two genes in the Z chromosome have to do with melanogenesis, TRPM3, and the OXCT1 has to do with carotenoid metabolism. Now, among all the peaks here for the outliers, the one that stood out the most is the one in chromosome 24 that includes the gene BCO2. So what does BCO2 do? It's a protein that encodes for the beta-carotene oxygenase 2 protein, and it does oxidative cleavage of carotenoids. BCO2 is found in the inner mitochondrial membrane and has a broader substrate specificity than its relative BCO1, which is expressed in the cytoplasm. BCO2 breaks down beta-carotene with obviously the yellow color and produce end products that are colorless. So to illustrate the functions of BCO2, this petri dish is an in vitro functional assay of BCO2 expressed in E. coli. So there are two versions of BCO2 here, the human and the mouse. The human BCO2 is 40 times weaker than that of the mouse. That's why in humans we could accumulate pigments in the macular region of our retina. With the mouse BCO2, the Z-xanthine, which is a beta-carotene, produced by the E. coli cell is rendered colorless compared to the negative control and the E. coli with expressing human BCO2. Here's a zoom-in view of the genomic region around BCO2. We have selected SNPs that are differentiated between parental candii, represented by these two birds with the yellow genotypes, and Villinus, the birds here with the red genotypes. Now, if you look at the golden collet candii here, you can see that each individual has an introgress block coming from Villinus, meaning they have the red genotypes, alleles coming from Villinus. This block starts upstream of the BCO2 gene and ends downstream of the gene with a total size of around 26,000 base pair. If you look at the genotypes of the normal candii from west of the river, we can actually see two birds that are heterozygous for the BCO2 gene. So meaning they have orange genotype instead of yellow genotype. So that tells us that each bird here has one haplotype from parental candii and one haplotype from Villinus. So when the specimens are actually inspected, these two heterozygous individuals actually has some yellow coloration expressed in the neck and the back feathers. Now compare that to a photo of a normal candii with white in the back and the neck. Next, instead of just looking at individual genes, we wanted to look into whether any genetic or signaling pathways as a whole are enriched for outlier loci. So for this, we use pathways and their constituent genes classified by the Panther classification system. There are a total of 275 outlier windows supported by one or more of the metrics as shown before. And there are about 300 plus genes inside or within 20 kb of these outlier windows. Now, the vast majority of these genes that fell close to an outlier window are from 5 different pathways, and these 5 pathways are significantly enriched for genes close to outlier windows. The 2 most significant pathways have something to do with angiogenesis or the development of blood vessels. The third pathway is the integrin signaling pathway. Proteins in this pathway are involved in adhesion of cells to the extracellular matrix. And finally, we have 2 pathways related to gut functions and stimulation of gonad activities. Now, finally, to validate the patterns we saw with whole genome sequencing, we decided to sample more broadly across a hybrid zone, but sequence fewer genetic loci using rat sequencing. We sequenced around 20,000 SNPs from 70 plus individuals from this population. So to your right is a plot of the hybrid index of birds from population arranged from east to west. So Villinus in the east and candii from the west. Birds that are genetically Villinus has a hybrid index of 0, and pure candii has an index of 1. As you would expect, the golden-collared candii represented by these populations are genetically candii with a hybrid index of close to 90%. The true hybrid zone is actually shown by the red arrow in the map here between the golden-collared candii and Villinus, and this red arrow here in the plot. Individuals in the hybrid zone has a hybrid index of between 10 to 60%. So using this data, we wanted to find out if Villinus alleles are indeed introgressing into golden-collared candii using a genomic cline approach. So if an allele from Villinus is not introgressing into golden-collared candii, we expect the genomic cline of a SNP to look pretty much like the genome-wide average, as shown in the plot on the left. Now, if an allele from a Villinus is introgressing, we expect the cline to look like the plot to the right. So where the hybrids are birds that are not 0 or 1 in their hybrid index. If an allele from Villinus is introgressing, we expect the cline to have this shape where the hybrids have a high level of Villinus ancestry compared to the genome-wide average. In other words, this SNP here has a shifted cline center. We have identified 120 SNPs from rat sequencing that are within outliers or up to 10 kb base pair away. And statistically, these SNPs are more likely to have significantly shifted cline than SNPs from the rest of the genome. So out of 122, 18% have shifted cline compared to the genome-wide average of only 10.4%. So in summary, melanin in the barbules and lutein in bark explain color differences in manakins. So by mixing and matching the amount of lutein and melanin deposited in different plumage tracts, a variety of plumage patterns can be created in the genus. We are able to pinpoint candidate genes that are responsible for color differences. So these genes either show signature of introgression into golden colored candida from villiners or are differentiated between normal and golden colored candida. Now, the genes we found have a range of functions, but that also include melanin and carotenoid metabolism. Now, changes in cis regulatory region of BCO2 we think allows for tissue and sex-specific activation of BCO2. This is why we think a bird can have different colors in different plumage tracts, and also color differences between males and females of the same species. And finally, a number of pathways are enriched for genes near outlier windows. So the third most important pathway, the integrin pathway, actually works hand in hand with the angiogenesis pathway, the two most important ones, to trigger endothelial cell survival, proliferation, and organization into blood vessels. We believe that this process has something to do with barbule growth or the deposit of melanin or carotenoid in the feather itself. The gut function pathway is actually really interesting, and we believe that it may have something to do with the absorption of dietary carotenoids because these compounds are lipophilic. So I want to thank you for your attention and acknowledge this funding sources and the support from this institution and various people. And I'll be happy to take any questions.